Hello, and thank you for joining today's webinar, High Sensitivity TruSight Oncology 500 or TSO500, Detection of Somatic Variants in Matched FFPE and Plasma, Implications for Liquid Biopsy Biomarker Evaluations, presented by Discovery Life Sciences and moderated by Scientist.com. Just a few reminders before we begin the presentation. This webinar is being recorded and will be made available to all registrants and attendees within 48 hours after the event. As we are all continuing to work from home due to the pandemic, we hope you will understand if we run into any technical issues or hear background noise from the speakers. We appreciate your patience and your involvement. If you have any questions during the presentation, please ask them by using the Q&A function, which you should see at the bottom of your Ring Central window with the large letters Q&A. Our panelists will answer any submitted questions at the end of the presentation. Before diving into today's presentation, I'd like to introduce myself and our panelists. My name is Maria Menicu, and I'm the Scientist.com Category Director for Human Biological Samples. I have worked for the UK's National Healthcare Service, managing the liver transplant service for one of London's transplant centres. I was also part of the founding team of UCL's initiative to enable ethical and compliant access to fresh human tissue for research. Now, I'd like to introduce today's speaker, Dr. Shawn Levy. Dr. Levy is the Chief Scientific Officer of Discovery Life Sciences Sequencing and Bioinformatics Laboratory, HudsonAlpha Discovery. He received his PhD in biochemistry and completed a postdoctoral fellowship in genetics in 2000 at Emory University in Atlanta. He is a faculty investigator and the founding director of the Genomic Services Laboratory at the HudsonAlpha Institute for Biotechnology. Before joining HudsonAlpha in 2009, Dr. Levy was a faculty member at Vanderbilt University Medical Center and the founding director of the Vanderbilt Microarray Shared Resource. Dr. Levy focuses on the development and application of genomic and bioinformatic technologies and methods to support scientific investigation. His team uses high-performance genotyping and sequencing technologies to support translational and clinical-based projects. Joining Dr. Levy today during the Q&A session will be Dr. Nipresh Prasad, Director of Feasibility at Discovery Life Sciences. Thank you, Dr. Prasad, for being with us today. Now, I'd like to hand it over to Dr. Levy to begin his presentation.
All right, thank you, Maria, and thanks to everyone attending today to hear some of our recent data utilizing the Illumina's TSO500 circulating tumor DNA profiling panel, as well as the TSO500 high throughput assay in its application in primarily some controlled experiments to really define its range of operating parameters in our clinical and translational lab, as well as investigation into some actual samples. So today's presentation will follow the overview that you see on the screen. We just had the introduction from Maria and the Scientist.com team. The remainder of the presentation will focus on a few key points that touches on both sample extraction and sample quality and the impacts of input material on resolution in the types of comprehensive profiling assays that we'll be talking about. Some considerations for the extraction of cell-free DNA from plasma samples. An introduction to and covering some of the basic details around the TSO panels that I just mentioned. And then finally looking at some comparative analysis of paired samples from both plasma and FFPE from the same patient, and then reviewing some of the opportunities for interpretation of detected variants through tools like PierianDx, which produces an annotated PDF report. And then finally, we'll have some conclusions, and then a couple of brief slides around more of what our laboratory does and capabilities. So a little bit of a reverse presentation in that sense. Rather than spending too much time on kind of how our lab works and what it does, I've saved that for the end, in favor of diving right into some of the materials around the TSO500 on both circulating tumor as well as FFPE material. So to orient everyone to some of the core biology that we'll be discussing, this is a little bit of a wordy slide, but knowing that everybody's reading it on their screen, wanted to touch on a couple of high points. I'm sure we all appreciate that tumor biopsy has really become a standard method for molecular cancer characterization and diagnosis. So when you combine resolution that can be gained from a tumor biopsy with the rapidly advancing imaging capabilities, we're approaching a time of being robustly able to detect very early cancer. And then with very targeted therapies and therapies that are unique, almost unique to a particular type of cancer or a particular progression of cancer, understanding the molecular underpinnings of that disease is becoming critically important. Fixed formalin paraffin embedded samples still represent the major sample type for most cancers, and that's understandable given the reliance on pathology and the overall historical nature of how cancer remains diagnosed. But liquid biopsies, the idea that you can detect shed nucleic acid material in body fluids, with blood being the primary sample, but certainly not the only sample that circulating tumor DNA can be detected. It's been robustly detected in urine and other sample types as well. But essentially, cell-free DNA is fragmented DNA that is found in all non-cellular blood components of healthy individuals. And then in the presence of cancer, a fraction of that cell-free DNA can be detected that has been shed by circulating or from tumor materials, commonly referred to as circulating tumor DNA or ctDNA. So stated a little differently, all circulating tumor DNA is cell-free DNA, but not all cell-free DNA is circulating tumor DNA. The advantage of these detection is that the profiling is non-invasive, convenient, and as the technologies move forward, may be quite comprehensive, not only for early detection of the disease, but also for very robust monitoring of residual or recurrent disease. So circulating tumor DNA is, generally speaking, between 150 and 200 base pair fragments of DNA that are released by tumor cells into the bloodstream, and they represent a small fraction of the total cell-free DNA that is present. The normal half-life of circulating tumor DNA is less than an hour, which suggests that it can reflect dynamic tumor characteristics during treatment and during disease. This also implies significant considerations for sample collection and sample processing as well that we'll touch on in a few moments. So circulating tumor DNA has also been proposed to have multiple origins and commonly not derived from a single source. And 3 primary sources are listed on the slide: apoptotic or necrotic tumor cells, active tumor cells, or actually circulating tumor cells themselves, where there's whole cell material circulating in the blood from tumors. Obviously, the site of the disease, the type of cancer, and the progression of that disease would influence which of the listed sources or perhaps even other considerations or other hypotheses may come into play. So this is certainly not an exhaustive list, nor meant to be a highly specific collection of precisely how circulating tumor DNA can get into the blood. But because the genetic information carried by the circulating tumor DNA is the same as the tumor cells, and it is present in the peripheral blood, it's an attractive option for molecular detection of cancer or residual disease that, again, is very non-invasive when contrasted to a biopsy, which can be very disruptive to the site of disease, and in fact can even increase the chance of metastasis or alter the progression of the disease. So the quick notation on extraction of DNA and RNA from FFPE samples. And this is something that we've worked on for a number of years, and won't spend a tremendous amount of time on this other than to describe a conclusion that was developed, work primarily led by my colleague Nirpesh Prasad, who's joining me for the presentation today and will be available for the question and answer session. And that is very, very simple, that a significant amount of intact RNA is in fact present in the majority of FFPE samples. And it is through that RNA is often degraded during the extraction process. So if the extraction process is modified to allow a very thorough but gentle extraction and retention of that higher molecular weight RNA material, you can significantly change the quality control and subsequent use of that material in downstream assays. And what you're looking at here is a comparison of that. So on the left-hand side of the upper panel of the slide, where it says standard protocol, where 6 of 79 samples had a DV200 of greater than 20%. The DV200 is a metric of what percentage of the RNA molecules were 200 nucleotides or larger. And so you see 6 of those 79 were above that DV200 of 20%. contrasting to the right-hand panel of the same slide, where 90 of 90 samples had a DB200 of greater than 20%. And you can even see that a significant fraction of those samples were in excess of 50% DB200, and even a subset greater than 80% DB200. So this was a variety of tissue types, a variety of cancer types. And the point being that the key variable, it was not necessarily the tissue type, but in fact, many of these samples were matched, meaning a fraction of the tissue in the standard protocol and then the same tissue in the HudsonAlpha Discovery protocol. If we instead look at the same comparison, but now dividing the data by disease type, which is shown on the bottom panel with the blue and the orange bars, blue or purple being the standard protocol, and then the orange being the HudsonAlpha Discovery protocol. And the same, in this case, a gray line indicating a DV200 of 20% or more. What you see is there is a tissue-specific relationship in terms of the magnitude of change between the protocols and even the overall DV200 that you see for a particular type of tissue. Notice that prostate and ovarian are two examples where you see a fairly dramatic change between the two protocols, whereas breast tumor, for example, or even a skin tumor, not quite as dramatic. And this has been reasonably reproducible, but there are a number of variables, sample processing time, embedding essentially all of the tissue handling considerations that happen prior to the FFPE sample, and even the size of the block, how it's stored, the age of the block, all have some influence. But the takeaway message, though, is very, very consistent that the isolation method is a key determinant in terms of how much of the intact RNA can be recovered. So all of the data that we're showing from FFPE material in the remainder of the presentation was isolated via the methodology, the HudsonAlpha Discovery methodology that was just described. If we also look at the extraction conditions around cell-free DNA, or in this case, the extraction of cell-free DNA with the goal of detecting circulating tumor DNA. There were two primary kits that were looked at, one being the QIAsymphony cfDNA kit, and the other being the Apostle Minimax kit from Beckman. And you can see some of the key metrics of the kits. The number of samples that were compared was 24. The time for extraction is very equivalent, approximately 2 hours. Both are automated protocols. The QIAGEN kit is automated on the QIAsymphony platform. The Apostle Minimax kit is automated on the Beckman i7 platform. And so in our laboratory, we have both instruments, so both of these were run in an automated fashion. The key difference is highlighted in red in row 2 under the sample volume. So the QIAsymphony kit uses 4 mils of plasma input, while the Apostle Minimax kit uses a single milliliter. Now, there's certainly some options for alterations of that, but the listed conditions is how the kits were run in this particular case. And then what you see in the bottom left panel is the same samples, which are indicated by a de-identified patient ID indicated on the sample numbers, which starts with a 6158. So the specific identification of those isn't particularly important. But note that the number of samples that had a yield of less than 30 nanograms, which 30 nanograms is a key input metric to be at or above for the subsequent assays. 16 of the 24 samples had yields below 30 nanograms for the QIAGEN protocol, and 8 of 24 had yield of less than 30 nanograms for the Apostle Minimax. But keep in mind, it was 4 times the input in the QIAGEN protocol compared to the Minimax. And so if we were to normalize input amounts, for example, using 4 1 mil extractions for the Apostle, you can generate significantly more amounts of cell-free DNA from those extractions. So based on these results, the remainder of the assays and the remainder of the analysis that we'll be discussing used cell-free DNA that was isolated with the Apostle Minimax kit for the data that you'll see in a few moments. So very brief, this information is all very well annotated and very well organized on Illumina's website. But for those of you not familiar with the TruSight Oncology panel, this is some of the highlights. That it's a pan-cancer analysis platform that uses a target enrichment to allow targeted DNA sequencing and targeted RNA sequencing. In the case of the high throughput platform, the circulating tumor DNA platform looks only at DNA. Obviously, we're isolating only DNA from the circulating tumor or the cell-free DNA fraction, so there's no reason to look at the RNA in that particular fraction. However, in the FFPE material, we're isolating both DNA and RNA from the same sample and looking at both fractions in a targeted basis. The specialized sample types are listed as appropriate. This is a human-based assay, and then all of the sequencing was performed on the NovaSeq 6000. The variant classes are listed, and they're the usual suspects in this kind of work: copy number variations, gene fusions, insertion deletions, and of course single nucleotide variants, and as well as an ability, depending on depth of sequencing, to analyze various percentages of somatic variants, which we'll talk about as well. So I'll be very brief on this particular slide. This is just to indicate that the TruSight Oncology as a system has a comprehensive set of recommendations from Illumina, from the coverage of the panel to the input material through to how to sequence it, analysis options via the Dragon platform, and finally, in partnership with PierianDx, tertiary analysis for interpretation and reporting. So there's two general workflows for the circulating tumor assay, and that is a standard workflow which uses an S2 or an S4 flow cell at a 2x151 run configuration and a paired end sequencing. There's an 8 base pair dual index, which is required. And so if you have enough samples to address lane loading, it does require an XP workflow on the S4 flow cells. The circulating tumor or cell-free DNA-based assay does require a high depth of sequencing. It's about the equivalent of a human genome. Or a whole human genome, so approximately 24 samples per S4 flow cell, generating around 100 to 120 gigabases of data per sample is the recommended amount. So very deep sequencing for the circulating tumor DNA. And so the workflow looks like this. It's performed over 2 days. Again, won't go into this in a lot of detail other than indicate robustly available on Illumina's website, and only to highlight one change. So steps 7 and 8 shown in the red box, which is a quantification of the libraries, followed by a normalization of the libraries using some of Illumina's method. We skipped those 2 steps in favor of a Roche Kappa qPCR assay, and then a more manual normalization of the libraries using methods that we had in the laboratory. So that is the key, really the only key difference from the standard workflow was really just how we normalized the libraries and pooled them together for sequencing. So the cell-free DNA requirements, general guidelines highlight the key takeaways. Heparin tubes really cannot be used or should not be used. EDTA tubes and Streck tubes are preferred, with perhaps a bit of an advantage to the Streck tubes in a number of publications to help prevent hemolysis of the samples. And as noted earlier in the presentation, plasma samples should be collected and separated quickly after collection. Long separation times appear to have an impact most directly on tumor mutation burden calculation. So you see a change in the diversity and a change in the information content of the cell-free DNA with long collection times that are most robustly seen with an artificial decrease in TMB calculations. Double spun methods are very effective for collection of plasma out of EDTA tubes. 1 to 2 mils of plasma with no signs of hemolysis, immediately frozen at -80, make for very strong input material, and you absolutely want to avoid freeze-thaw cycles before analysis. So the cell-free DNA input for the Illumina assay is greater than or equal to 30 nanograms in a 50 microliter volume, and then we generally do a QC qualification with a review of the isolated material on an adjuvant fragment analyzer or an equivalent device. So the data that we'll be showing you in a few moments was run on the following sample types, and we restricted it to these sample types in the interest of time for today to cover some key controls: the Horizon OncoSpan cell-free DNA, which was run in duplicate; the Seraseq ctDNA Complete Mutation Mix, which has a variety of either wild type or different allele frequencies that were also run in duplicate. And you can see the description of each of those control materials in the text. The Horizon has it's a cell line derived reference standard with a little over 375 variants across 152 key cancer genes, and that includes a number of different variant classes, including 27 indels, 21 deletions, 6 insertions ranging from 1 to 15 base pairs, And then the variants are present at a variety of allele frequencies. The Seraseq is a little bit different in that it has 5%, 2.5%, and 1% allele frequencies, 25 unique multiplexed variants that are in 16 genes that also cover single nucleotide variants, indels, but in this case, there are a couple of CNVs and SVs in the purified and plasma-like, and that's available as a purified or a plasma-like matrix format, so you can go through isolation. And then finally, a titration experiment where we had plasma cell-free DNA from a colorectal patient using a variety of inputs, 30 nanograms, 20 nanograms, and 10 nanograms were tested to show you the impact of input amount on resolution. So in this case, this is just some accounting of both the final library profile shown on the bottom, as well as the final library concentrations, insert sizes, and then the number of reads that were generated. Just highlight, note on the right-hand side of the slide, the percent Q30s and the Q scores were all very consistent with the different samples. Libraries were fairly indistinguishable from in terms of yields and sizes, et cetera, for the circulating tumor DNA samples. Draw your attention to the highlighted numbers here, and here is probably the most important and differentiating metric on the key metrics that you see. So that essentially, you can summarize this slide as you go down in input, you'll lose resolution in terms of coverage. So if you look at the percent of the exons that are in the panel covered at 500x versus 1500x, with a 10 nanogram input, very modest coverage. Even though the percent enrichment and the percent of usable UMI reads remains indistinguishable from the other samples, you see a significant decrease in the level of coverage. And as you go up to 20 nanograms input, it improves significantly to the 500x fraction. And then as you get to 30 nanograms and above, you see a significant improvement in the, certainly in the percent at 500x, and still seeing growing amount of coverage at the 1500x cutoff as far as percent exons. So this is the main reason why, and certainly verification of the input amounts, and the input amounts are important for overall resolution and the final both allele frequencies as well as accurate detection of variants. So if we ask the question, what is the reproducibility of replicates on a complex sample such as the Horizon OncoSpan, you can see that the replicate 1 versus replicate 2, both in terms of fold change observed in copy number variant genes, which you see on the lower left-hand side of the slide, which include EGFR, CDK6, MET, BRAF, and MYC, and then as well as in the detection of MSI sites, and finally the calculation of tumor mutation burden, which is shown on the upper left-hand side of the slide. All very, very consistent between the two replicates, and then if you review the allele frequencies and plot them as you see on the right-hand side of the slide, you see an R-squared number of 0.9989. So very, very consistent of replicates between the same sample through the assay. So using the same DNA sample, we can very robustly get the same key overall metrics such as TMB and MSI, as was mentioned, as well as copy number variants shown on the bottom. If we look at the Seraseq circulating tumor DNA complete mutation mix, now with specific allele frequencies that are annotated, which is 5%, 2.5%, and 1%, as well as the wild type that is present. Not surprisingly, the wild type is not available for any of the given mutations that are assayed, because again, it's wild type. But then we ask the specifications for the control mix are the listed allele frequencies, 5, 2.5, and 1%. And then what's shown in the panel for each of the given mutations that have those allele frequencies, it's what were they detected and reported at in the assay. So you see for the 5% fraction, the majority were slightly under 5%, some right at 5%, and then a small number that were actually detected at slightly over 5%, both for SNVs as well as the deletions and insertions. The CNVs and the gene fusions were simply detected as present or absence, so indicated if they were detected as a yes, and if they were not detected as a no. And then, again, pretty consistent performance as you go from 5% down to 2.5%, and then finally down to 1%. And this, again, is in the Seraseq control. So far, or I should note that 100% of the expected SNVs, deletions, insertions, and gene fusions were detected in all of the mutation mix samples. So this is what you're looking at here is a representative of one sample, but again, this is highly representative of the data from the other samples. If we looked at a colorectal cancer adenocarcinoma, So again, instead of a control sample, now stepping into a real patient sample and ask some of the same questions. What is the effective input amount? We see a replication of what we saw in the control samples, as you see here. If we also then ask what is the effect of reproducibility among replicates, you can see that the reproducibility gets better as you go up in the input material. And this is really the case for most of the samples, or I say most of the allele frequencies, but as expected, the allele frequencies in the middle to lower end of the range become more reproducible as you go up in input amount. So again, more data to really hammer home the point that for the cell-free DNA assay, with the specific goal of detecting some of these circulating tumor markers, 30 nanograms or more of input is a very important benchmark. So shifting gears a bit to the high throughput assay, we can look at it as very similar in workflow as to the cell-free, different sequencing conditions, because you don't need quite as much data. And then of course the inclusion of the RNA fraction as part of the preparative steps to allow detection of fusions and splice variants in the assay. So there's two workflow options that are shown here. You can do a DNA only workflow or a DNA and RNA. All of the data that we'll be showing was performed in the dual workflow. So the DNA and RNA workflow, as you see here. So the way that works essentially is you do a synthesis of cDNA off of the RNA fraction, a fragmentation of the DNA, and then those libraries essentially are processed in parallel, as shown here. There's the inclusion of a UMI indexed adapter. So there is a unique molecular index that is included. Because this NovaSeq sequencing platform was used, there is the inclusion of unique dual indexes. And then similar to the cell-free, the same alteration in the workflow where instead of normalizing and preparing for sequencing per the Illumina protocol, we utilized a kappa qPCR and then normalized the libraries via our laboratory methods. But again, a very mild departure from that. So FFPE requirements are shown here. They're probably about what you'd expect. 3 10-micron sections of greater than 50% tumor cells, less than 20% necrosis, with an approximate tissue area of 50 millimeters squared is ideal. If only slides are available, 8 to 10 non-stained slides would be helpful. If frozen tissue, about 30 milligrams of flash frozen tissue. And as far as for sorted cell sample, say for example from multiple myeloma patient, 1 to 2 million cells is ideal. So of any of the conditions you see listed, we generally see very robust both DNA and RNA yields in a dual extraction methodology to allow the metrics of the downstream assays to be met very, very well for the vast majority of samples. Similar to the earlier design, 2 specific controls were used, both the Horizon, in this case, the OncoSpan genomic DNA control was run in duplicate. This is also a cell line derived sample that contains the same variants that you heard about for this circulating tumor version of it. And then the same in the case of Seraseq, this was an FFPE mock control, which had a number of NTRK fusions in the RNA sample. And then there was a titration experiment of both DNA and RNA from a single colorectal cancer patient. 80, 40, and 20 nanograms of FFPE DNA and RNA input were tested, and then 10, 20, and 30 nanogram input from the ctDNA from the matched plasma from the same patient, basically the same data that you were seeing a few moments ago. So if we look at the reproducibility in the OncoSpan genomic DNA reference, you see reasonably good, but not quite as good as the circulating tumor assay, and both the allele frequency for the genomic DNA, and then on the repeat run on the other side, which will run in duplicate at a 40 nanogram input. The difference between the 2 plots is the plot on the left is comparison within a run, and then the plot on the right is the comparison of the 2 samples between 2 independent runs. So essentially, really no difference between the same sample that is compared within a single run or a sample that was run in 2 different batches. If you look at the comparison of the Horizon circulating tumor DNA and the genomic DNA OncoSpan controls. So in this case, now combining the data from the circulating tumor-based assay and then the genomic DNA-based assay, and you can see the expected allele frequency for a subset of the mutations shown on the left-hand side of the slide. and then a correlation between the allele frequency detected in the genomic DNA fraction versus the circulating tumor DNA fraction for the Horizon OncoSpan controls. Again, recall the Horizon controls have the same expected allele frequency, but one in a circulating tumor DNA format and the other in a genomic DNA format. And you see quite comparative results between the circulating tumor DNA shown in the second to last column and the genomic DNA shown in the last column, with one exception of an EGFR mutation that was at an expected allele frequency of 1%. It was detected at 0.7% in the circulating tumor DNA. When undetected in the genomic DNA, the primary reason for this is likely sequencing depth, in the sense that the sequencing depth on the circulating tumor is very, very deep in contrast to the genomic DNA assay, and that's the most likely reason why that variant was missed in the genomic DNA fraction. So if there is interest in very, very low frequency variants in a genomic DNA-based analysis, increasing the sequencing depth perhaps above the standard specification would be maybe recommended. So in the case of the Seraseq NTRK RNA fusion materials, here all 14 fusions were confirmed in both replicates within the same run, as well as between 2 independent batches. So essentially the detection of the specific control fusions went precisely as expected. Now, if we begin looking at the colorectal cancer adenocarcinoma, again, a titration experiment of 80, 40, and 20 nanograms input, and then start asking what are the changes in variant resolution, and that's indicated on the right-hand side of the slide under the overlapping variants column header. So what this means is, with 80 nanograms of input, there were 626 variants detected in that sample. The same sample with 40 nanograms of input, 613 of the 626 variants were detected. And then finally, with 20 nanogram input, only 96 out of the 626, or 613 out of the subsequent 626 variants was detected. So again, similar to what was observed in the earlier work, and again, and also indicated on the DNA metrics panel on the left-hand side of the slide. I'll draw your attention to the percent exons at 100x. Notice it goes at 80 nanograms, it's almost 98%, drops to 34% at 40 nanograms, and essentially at 0 at 20 nanograms. So the impact of decreasing amounts of input material is fairly significant when viewed both from a coverage perspective, shown on the left-hand side of the slide, as well as a variant calling performance perspective, shown on the right-hand side of the slide. You'll also see similar effects in the review of both TMB calculations as well as MSI calculations, which are shown on the middle and lower left-hand side. And then just the pie chart essentially just describes the different variant classes that were detected. So if we then look at the matched plasma and FFPE comparison. So in this case, the colorectal cancer adenocarcinoma. So again, a real patient sample, looking at the circulating tumor DNA analysis, and then pairing that with the TSO500 high throughput DNA analysis. So obviously 2 independent runs, high throughput DNA performed as described earlier, circulating tumor DNA performed as described earlier, and in both cases using the recommended input amounts of material, 80 nanograms on the circulating tumor, the TSO500 HT, 30 nanogram input for the circulating tumor. And note that for total variants, again, the FFPE fraction, 626 variants were detected in the plasma, 622. If we ask what the overlapping variants there, so 573 variants were found. Of the 622 variants detected in plasma, 573 were found in the FFPE. And then of the 626 variants detected in the FFPE, 593 of them were detected in the plasma. There were a number of unique variants found in each fraction, 27 in FFPE, and then 29 in the plasma. And there'll be some mention of those differences in a moment. We look at tumor mutation burden shown on the right-hand side of the slide between the matched FFPE and plasma samples. Genomic DNA had a TMB score of 8.7. We saw a score of 4.9 in the cell-free DNA fraction. If we looked at the coding region, you can see some of the differences in the number of passing eligible variants shown in each one. And again, this is on a per megabase basis. MSI characterization is also shown. And then finally, if we look at, in the case of gene amplifications, splice variants, and fusions, in this particular sample from this colorectal cancer adenocarcinoma patient, none were detected. So that's why each of those is indicated as NA. There were a couple of interesting differences between the FFPE, where you see the allele frequencies listed here for both an APC mutation as well as a KRAS mutation, found at 100% as a synonymous variant in this particular case in both the plasma as well as the FFPE for this the APC gene is a key tumor suppressor gene whose protein functions as a key antagonist of the Wnt signaling pathway, and it's one of the most frequently mutated genes and known as a driver gene in colorectal cancer. So not at all a surprise to see this gene there, and again detected at 100% allele frequency in both fractions. Interestingly, there's a KRAS gene which was found in the FFPE sample but not detected in plasma as a missense variant. In this particular sample, indicating some of the value of looking at both. If we delve into some of the variants that are unique in the genomic DNA, variants that are unique in the genomic DNA from the colorectal cancer patient, you see some of those. Some key ones are highlighted here, where we see out of the FFPE material, about 20, the allele frequencies shown on the second to last column, and then for the same variant, what we saw in the cell-free DNA. So, in some cases, fairly significant allele frequencies that were just not detected in the cell-free portion. So, still exploring and validating some of these, but this approximate magnitude of differences between the genomic DNA fraction and the cell-free fraction is fairly typical. If we ask the reverse question, what are the variants unique in the circulating tumor DNA that were not present in the genomic DNA? Again, there is a few potentially interesting variants that are also shown here. Note that many of them have very low allele frequencies. There's 3 or so that had higher, the MAP3K14, a notch 4, and a PRKDC that had higher allele frequencies, which are a couple of frame shifts and a missense variant. Again, validation work continues, but for the most part, these are indicative of some of the unique variants that are often seen when comparing a circulating tumor versus a matched FFPE material. Variety of reasons that these can happen. A couple of notes about some of the genes that are highlighted, both ATM as well as the ATX, or in the TET2, that is that may have some biology behind them, but again, these are should be looked at in a more hypothesis generating basis at this time. So if we then take the next step of after generating all the data and then going through an annotated report, just to give you a flavor of what this looks like, you can kind of see for the same patient, looking at the matched plasma and FFPE samples, where here on the left-hand side, you have the TruSight Oncology 500 circulating tumor DNA assay, and then the Oncology 500 high throughput assay. In the case of this KRAS mutation that's highlighted in box, that mutation was independently identified or independently verified by an orthologous assay. So it is a good finding. It is appropriate. And then again, we see the highlighting of many of the same variants that you've seen before, but now they are characterized into different tiers of characterization, and then indicated with in terms of tier 1, using the example shown on the right-hand side of the slide out of the TSO500. This is any variants which have a strong clinical significance. Obviously, I won't go through and read all of this text, but you can see that in the report is a significant amount of both summarized clinical impact shown at the top, and then a much more detailed and significantly referenced interpretation of that particular variant shown on the bottom. And then you see a similar shown on the left-hand side of the slide for, in this case, a tier 2 variant, which means it has potential clinical significance. Same formatting for the report. And this continues on for any of the variants in a similar format in the different tiers. And then, of course, the variant also has a significant amount of value in terms of potential clinical trials, indicating that on a report by report basis, if there's an appropriate variant where there is an appropriate trial, the phase, the trial identifier, and the title of the trial is all provided as part of the report. So a very convenient way to go from fairly high density data across hundreds of genes into a very interpretable and robust report on a per patient basis. And then finally, this summarizes some of the classification levels of evidence in a little more detail, all very available on Pierian's, both Pierian information from Pierian as well as Illumina that can be looked at. Then finally, last couple of slides. These are some additional samples that were run. You can kind of see some of these metrics that are shown across the top. And this was all on the TSO500 HT version. Some example: thyroid, colorectal, endometrial, and gastric cancer. You can see again fairly similar metrics as you scan down, and then but you do start seeing some very disease-specific phenomenon. For example, in the what genes are detected as amplified, whether or not splice variants were detected, or whether or not fusions were detected. So essentially, the assay functioning exactly as expected across a diversity of patient and sample types, with very fortunately with very, very similar technical metrics for the QC evaluation of the process, and then leading to rapidly divergent results depending on the actual mutations and different variant classes that are detected. Same thing if we extend some of these reports into those patients. Again, you see the reports pulling up the expected findings, tying them to clinical trials, as you see on the right-hand side as well as the left-hand side, either for ETV6 or NTRK3. In this case, the fusion transcripts, which has 2 clinical trials. This was found in the thyroid cancer sample, and then in the genomic findings shown on the other, and ERB2, which was found in the endometrial carcinoma in this case. So again, the findings in these patients replicating the expected biology. So last slide for the data content, kind of just drawing some conclusions from all of this. We see a very strong correlation between replicate samples in each assay. So that's is very encouraging and important, I think, to given that many of these assays or many of the goals of most of the studies that use this assay is to be able to assess a range of allele frequencies. Therefore, the ability to accurately and reproducibly detect across a range of allele frequencies in a reliable and repeatable manner, both within the same run as well as between independent batches, is critically important. We see a strong relationship between coverage and sample input material, and a correlation between, and also strong correlations between the circulating tumor DNA and genomic DNA from the same sample, whether that sample was controls or that sample was a unique patient sample. And in the case of the patient samples, there are also unique findings which may contain some interesting biology. I think these data provide further impetus towards the incorporation of this methodology into routine clinical care in the management of advanced solid tumors, such as metastatic colorectal cancer, which you saw in one example. Obviously, the molecular resolution that this brings, and then the capability of doing large batch sizes as well as high consistency and the possibility of fast turnaround times certainly allows the utility of the assay to be very broad. We found the PierianDx reports to be both convenient and comprehensive as an option for assay interpretation. And then, if there's further interest in discussing either any of the data, more of the background of these projects, or even extending through to pricing information, there's an email address shown on the screen. And then I'll just conclude with a couple of quick slides and acknowledgements of our genomic services extend well beyond what you saw today with TSO through a very comprehensive collection of short read and long read sequencing contexts. Here's some of the, we have a very large capacity on Illumina sequencing with 9 NovaSeqs. We're currently up to 4 PacBio Sequel 2s, a couple of Promethean, Oxford Nanopore Promethean systems. So we have a very comprehensive genomics center where we've even applied some of the higher molecular weight RNA isolations from the FFPE material, and even looked at IsoSeq on PacBio, for example, on some of these. So finally, even beyond genomics, Discovery Life Sciences has unique resources which allowed us to perform these kinds of studies as an internal development effort, where we have access to over 10 million research quality biospecimens, a strong bioinformatics team, an amazing cell services laboratory, which does flow cytometry, single cell 10x library, cell culture, custom biospecimen processing. And finally, we have colleagues on the IHC side of the business. So we're very, very fortunate to be able to perform these types of genomic analyses, in the context of a broader company that has, significant expertise, really rounding out and allowing us to look comprehensively at all of the variables that have been described today. So I'll acknowledge the laboratory team that performed the work, which you see here in the middle. Of particular note, the gentleman second from the left in the slide is Nipresh Prasad, who was responsible for essentially all of the work that we looked at today, as well as the development of the dual extraction assay. So he'll be joining me for the question and answer session. So I thank you for your attention and look forward to answering any questions that you have.
Excellent. Thank you so much, Dr. Levy. That was a very informative and interesting presentation. And I see that the audience also already has come up with some questions. Just a reminder to everyone, if you do have any questions, please ask them using the Q&A function, which you should be able to see at the bottom of your Ring Central window. I'll start off with a question that Dr. Prasad has kindly already provided a brief answer in the chat, but to help capture all the information for all our attendees. What is used to quantify the cell-free DNA adequately, and is a bioanalyzer quantification an alternate to fragment analyzer quantification for cell-free DNA?
Yeah, so we use both bioanalyzer and fragment analyzer just to cross compare, how the data looks like, running on different instruments. But yeah, we went with the bioanalyzer concentrations as recommended by Illumina.
Fantastic. Dr. Levy, is there anything you'd like to add?
No, I think as Nipresh said, we find them to be pretty functionally equivalent, and I think the biggest item to look for, especially across a diversity of samples, is divergence from from your typical observation. Divergence from the mean is the most important thing to look at on those profiles. Not necessarily exactly the slope of the line or precisely the height of the peak. I think there's a reasonable variance that can be observed there due to some of the differences in how those machines operate. But you just want to be sure you're comfortable with whatever QC mechanisms are being used so that it can be applied consistently.
Fantastic. There is something that sort of like follows on from that. During the validation of an in-house targeted next generation sequencing panel for cell-free DNA, how significant is it to have concordance in the VAF commercial controls, particularly the expected against the observed?
Well, so that's it's an interesting question, and so I guess providing a simple answer is. I believe the commercial controls can be very helpful to help define the sensitivity and specificity of a given assay. And if you see a lack of concordance either between different assays, between run conditions, or between controls, investigating that may be quite critical to ensure that whatever allele frequency detection sensitivities are being validated remain consistent and remain accurate as a variety of real world samples are applied. So stated that a little differently, the controls represent very clean, very consistently performing samples. And so if there's variance in the detected allele frequency versus expected, then that should cause some pause and a careful evaluation of as to how that would apply to a real world sample that has a number of additional variables that are introduced: tumor purity, percent necrosis, a variety of other impacts that you don't have in the control. So the controls really should represent the easy and the very accurate, highly reproducible. But I think you can at the same time use those controls as an assessment of where the boundaries may be on the overall assay, and then just scale or validate accordingly from there.
Fantastic. Another question is, what method was used for orthogonal testing?
So let me answer that. We used the pyrosequencing as well as the QIAGEN multimodal and independent NGS assay. So there were two different assays that were used to confirm those mutations, and on both of them they came back as was identified on the TSO.
Yeah. So it's so essentially one pyrosequencing assay and one independent NGS-based assay with a different amplicon design was used to validate.
Fantastic. How is the cheap gene detection in the circulating tumor DNA data? What is the false positive negative rate?
I'm not sure what the question is in referred to the chip gene detection, but I guess I would, as far as a false positive and negative rate, what we can say so far is in the controls, our false negative rate was essentially 0. We detected all of the expected variants that were characterized as part of the specifications of the control samples. Now, we are currently reanalyzing the data to ask, of all of the other nucleotide positions that are also detected in the sequencing assay, basically the surrounding sequence data. How consistent is that data? And we're just starting to look at that now. It seems very, very, very consistent, probably somewhere in the 99 point something range. But again, it remains under investigation. As far as false positive, false negative rates in the parental samples, again, in this case, we've shifted those gears a little bit, and we're looking at some deep exome and deep RNA-seq in samples that have also been run on the TSO panel. And what we can say, again, speaking broadly, is the additional coverage offered on the TSO500 panel, we actually probably see an enhanced coverage and therefore enhanced detection of lower frequency variants than we see on the exomes. So far, the false positive and false negative rates in the amount we've used this assay have been very, very low, at least as far as what we can detect.
Excellent. Another question is, when testing cell-free DNA samples from cancer patients, what is the best way to rule out variants arising from clonal hematopoiesis?
It's a great question. I guess it comes down to an experimental design question as to how much you want to rule out the clonal aspects of it. Yeah, I guess I think that would certainly be a bit of a broader discussion, in my opinion.
Correct. One more, slightly more technical question is, what sample types do you accept for the TSO500 high throughput? And if you get less DNA or RNA than the recommended amounts that you've mentioned, based on your titration experiments, will you still run the assay?
Yeah, it's an interesting question. So as far as the sample types, we fully appreciate the precious nature of all of these samples, and therefore we've tried to develop protocols and methodologies to essentially work with anything available, and then perform the isolations using optimal methods as we can either find in the literature or have optimized in the unique environment in our lab. Again, so not trying to represent that we have every answer to every problem, but I think what we've tried to do in the significant number of oncology, cancer and ongoing programs that we have, both in the pediatric as well as adult cancer space, is just try to have a collection of protocols that we know gives us the best possibility of analyzing these samples. So essentially, we're delighted to give anything a try. As I mentioned, between the biospecimen procurement, the cell services, and the immunohistochemistry groups that we have access to, we can often offer insights that are perhaps enabling. So that's just on the isolation side. Stated really simply, we're always up for a challenge in terms of sample type or sample amount. I think what we've shown in the presentation today, though, that there is a reality to the input amount, and the input amounts affect resolution. And I think we can then indicate that if 30 nanograms of cell-free DNA or 40, 50, 80 nanograms of genomic DNA are not available, then we just have to calibrate expectations around the types, the allele frequencies that are detectable, and the overall resolution of the assay that may be possible. So there is a plus minus there, in those cases. So we've have biased our work to try to have as high efficiency yields from those assays as possible. And then that's how we tailor some of the sample specifications. I did notice one other question in the chat. I'll just briefly around the Streck tube use. My comments about the rapid separation of the samples occurring as quickly as possible is really more related to the EDTA tubes, not the Streck tubes. The Streck tubes, just by their nature of how they're processed, are in fact significantly stable after processing. I should have been more clear on the comments about the, really the processing of the whole blood out of an EDTA tube is critically important to happen quickly, not necessarily out of the more specialized Streck tube.
Excellent. And we are actually coming up to time, so I think that completes our questions for today. Thank you so much, Dr. Levy and Dr. Prasad, for presenting and for all the valuable information you have provided today. If you have any additional questions or you would like to get in touch with us at scientist.com or the Discovery Life Sciences presenters, I believe the next slide has our contact information. And also this webinar will be recorded and will be made available to all the attendees and registrars within the next 48 hours or so you can listen to it again. Thank you all again for joining us, and we hope you found this presentation useful.